So the focus on my talk is really about looking at changes in protein interactions when a cell line becomes resistant to a particular therapy. Okay? So here's an example. Can you guys see them on your way? So in this case, this clinical trial was done at Cedars-Sinai about 15, 16 years ago. This is a female patient who had metastatic lung cancer. So if you look at the first image, you see that lesion on her lung, right? So this is a section taken this way. Right? So the heart is right here in the middle. Okay? She was given chemotherapy. There was no response. There was a trial at Cedars at that point to give 1 pill a day, this tyrosine kinase inhibitor, without any noticeable side effects. So this is pre-treatment. She was given the drug after a month and a half here, and 3 months later, you see the lesion is gone. So this is what we call objective response. That's what we want. So this is a success story. It turns out that if you look at the population of patients who are given this drug, is that a small proportion, is 14%, really have this dramatic response, and the rest don't. Okay. What we know now, which we didn't know later at that point, was that these patients ultimately would develop resistance. Okay. TK resistance is common. It happens anywhere from 3 months to about 2 years, but it's inevitable. It happens every time. Okay. So the key is that patients typically have 1 or 2 different types of activating mutations in the receptor. Right? There's a number of different ones, but this is the most common. And if you give, for instance, in this case, a drug that targets this receptor, you get this dramatic decrease of tumor burden. Over time, they become resistant, and in this case, the patient had the T790M mutation in EGFR, changed the affinity of the kinase, and therefore the drug was no longer effective, and they subsequently used chemotherapy, no drug, and without giving erlotinib for a period of about 1.5 years, when they redosed it, it actually has response. So this mutation came back. So it's a common paradigm. You give it a drug, there's no response, then you go back to chemo. Right? In this case, the patient was okay for about 1 year, and then you try back the first drug, and you get a response. In the second case, you always have a shorter period. So if the initial resistance period was 1 year, the second time you're not going to get 1 year. You're going to get much less than that. Okay. So what are the common known mechanism of resistance in these lung cancer patients? Right? So roughly half of them have this either acquired or developed resistant mutation in EGFR. It's called T790M. This is easy to detect because you could use PCR. You can pick it up. Another 5 to 10% have amplification of another receptor, c-Met, and it bypasses the shunt by the drug. So that axis continues to signal. There's a number of other mechanisms in small proportion, but you'll notice that roughly 30% of the resistant mechanisms haven't been characterized. So when we started this, we knew that there was an opportunity here, right? And the opportunity is, can we identify novel modes of resistance? The other thing in terms of doing proteomics is that how much change happens on a tissue when it becomes sensitive and goes to resistant? Is it a small change? Is it a big change? Is that change dependent on the type of resistant mechanism? So monitoring pathway dependence is kind of a biomarker story. A lot of times physicians give drugs, they don't even know what the target is. Chemotherapy hits every cell in the body. Is the effect because the tumors are growing slower? Is the effect that the stroma is changing? Are you changing the proportion of cancer stem cells? We don't know. We just look at tumor regression. So a lot of times we don't know what the target of the drug is, and if we do, we don't know how it responds. And ultimately for biomarkers, if you can find protein biomarkers that would indicate how dependent a cell is based on a signaling axis, then you could use that for biomarkers therapy response. So here's how we did it. So we use in vitro models. This is a lung cancer cell line that is hypersensitive to the drug. It has the same mutation that exists in those patient cohorts. Over a course of 6 months, we gave physiological doses of gefitinib, the tyrosine kinase inhibitor. Right? And over that time, those cells developed acquired resistance. We generally measured a 100-fold difference in sensitivity to the drug from the initial phase to the final phase. Okay? Next question is, how do we analyze and verify the resistant mechanism? Okay? So I'm summarizing a lot of years of work into one slide. So we started off with using phosphoantibody arrays. So we use this one by R&Ds. And we noticed that a group of, so here's the parental line on the left, and the three resistant lines are on the right. And you'll notice that this group is overexpressed or highly active in the resistant ones, while it's on the parental. We did Western blots to validate. So this was our initial cohort. Most of these are Sarc family kinases, and this one protein c-Jun. So in order to confirm that, we overexpressed these in a number of different lines. We measured the IC50s, and the one transcription factor, c-Jun, that gave us a dramatic change in IC50 to gefitinib was c-Jun. The other ones were marginal. When we do the reverse experiments where we knock it down, we see the reverse phenotype. We did this in a number of cell lines, and we were convinced that there's a link between increased expression and activity in c-Jun to response to EGFR-targeted drugs. Okay. So now we have a second hypothesis. Right? Since c-Jun is a transcription factor, we hypothesize that increased c-Jun would have a dramatic impact on the proteome. So we use quantitative proteomics, we use SILAC, we metabolically labeled the parental cells and the resistant cells, and we did a bunch of proteomics experiments, and we measured the change in the proteomic profile between sensitive and resistance. So I don't have the slide here to show you, but if I compare 10 different lung cancer lines with quantitative proteomics, and I measure the standard deviation in change from one reference to the other, generally, in terms of fold change, there's a 1.5-fold difference in average change in abundance when you look at all different cell lines. You would expect that. There's non-small cell, there's small cell, they have different phenotypes. They look different under pathology. When we looked at the parental lines and the resistant, the order of magnitude was also 3-fold. So going from sensitive to resistant looked a lot different on the proteomic scale than it does comparing a lung cancer cell line that is a small cell versus adenocarcinoma. So if we take our data and we use various pathway intercalators, in this case we use Go ontology, and if we look at the proteins that are associated with upregulation in the resistant lines, we see pathways that are involved in glycolysis, RNA modification, protein folding. Right? Proteins that are specifically downregulated are in RNA processing, regulation of apoptosis, protein biosynthesis. So if I use ingenuity pathway analysis, I put my proteins that are up and down, it also brings up c-Jun, the protein that I think is driving resistance. This is important because with mass spectrometry, typically you don't identify low abundant transcription factors. But based on the other proteins that were up and down regulated, this inference engine was able to kind of infer the existence of c-Jun here. Okay, so that gave us more confidence in this. Okay. So next, we hypothesized that the c-Jun interactome, the interacting partners for this transcription factor, would be different from the parental cell lines and the resistant ones. So how do you go and measure protein interaction? Well, I'm very practical. I don't say there's a right way. There's a very nice review. If you want to go and look, there's a number of different techniques you can use to look at protein interaction networks. These two hybrids have been around for years. So the two that we use is affinity purification coupled to mass spec and bio-ID. Okay. So briefly, affinity purification means that in this case, you have a protein of interest. This would be the bait. Right? And you would use antibodies directed to that protein. You want to do a pull down, and then you want to identify what you've done by mass spec. Very common. Challenge, once again, is how do you know is your antibody specific to the target? How do you know what's a contaminant protein? How do you know if what you're pulling down is specific to that interaction or it actually comes down part of a network? So you get part of the picture. Okay. The bottom technique, bio-ID, is fairly new. It was developed by Carl Rowe in 2012. It uses a protein from bacteria, BurA. Okay. BurA is a protein that in bacteria biotinylates substrates with a specific residue. It's very specific. If you make a mutation in that protein, it now biotinylates any substrates within 10 angstroms at a specific lysine. So it becomes a proximity sensor. So the advantage is that you would have to make a fusion protein with your bait, and you add biotin, and all the interacting partners, or I would say proximity proteins, will get specifically labeled with biotin. And then you do a direct affinity capture targeted to biotin to identify those proteins. Okay? I think these two methods are kind of work hand in hand. So we used both of them. The advantage of this one, you don't change the system. On this one, you make a fusion protein, and then you're able to interact, which are the ones that are in direct proximity. Okay. So how do we do this? So since we have access to metabolic labeling, we always do our pull downs with heavy labeled and light labeled cells. So for instance, if we're using an antibody to c-Jun, we'd grow those cells in heavy isotopic enriched media, and we would do our control with an IgG. And then we would use the ratio between the heavy and light as a way to get confidence that our pull down came from the heavy labeled or the c-Jun targeted. Okay? And we obviously swap labels. The key for that approach, as you guys can understand, is your reagents. So antibodies are notoriously dirty. We went through several dozen antibodies before we were convinced that our antibody was working. And BioID, you have to realize, is a fusion protein. Where do you put the fusion protein? Do you put it at N-terminus and C-terminus, right? We did both, to be sure. Okay? You have to optimize the biotinylation reaction. You do it typically 16 hours, how long it stays in that. So all these are details that if you're interested in this have to be optimized in your lab, in your setting. Okay? So contaminant proteins are a big problem for mass spec. So we do a number of different things to kind of reduce that number. Right? So first of all, negative controls. Have a construct that, for instance, expresses just BRA and not a fusion protein. So you get a list of proteins that are coming down just because of what BRA is interacting with. That's an important one. We use the same algorithm. This algorithm compares based on what you get from negative controls and your positive controls. It kind of gives you a likelihood of how realistic your proteins are. The CRAPOME is a database developed by Jean-Claude Gingras, University of Toronto, where they have compiled, I think, 400 proteomic experiments from various affinity purification experiments. And in there, you can see if the protein you're pulling down, if it's been pulled down in 500 other experiments, then most likely it's a contaminant. If you have a protein that looks like it's only been pulled down a few times with relative high abundance to the other ones, it gives you a higher likelihood that that protein is real. Okay? So we also do that. And if you're using BioID, keep in mind that in the mammalian systems, there's 4 or 5 proteins, mitochondrial proteins, that are endogenously biotinylated. So you have to take account for that. So we did that, right, 6 months worth of work, and this is what you get. Right? So in the middle is c-Jun, right? And I've highlighted the proteins that were specifically identified as interacting partners in the parental line are here. There's 18. Proteins that were specifically identified in the resistant lines, 79. Much bigger interactome. Yes?
What's your negative control for the BRA Jun cells?
Just BRA by itself.
But that has more protein than may even be in a different location. It's not in the nucleus and then you have to
So yeah, that's one of the things that you should do when you have a fusion protein is you look for immunofluorescence to see by the fusion protein changes localization.
But where is your BRA localized in that? In the cytoplasm?
Well, generally it's in the nucleus. In this case, c-Jun is hyperactive, and we find it at 75% in the nucleus, 25% in the cytoplasm. The BRA construct doesn't seem to change it depending on where you put it. If you put BRA by itself, it's not in the nucleus at all. So we don't have a control that looks at BRA in the nucleus by itself.
Are you trying it with another different transcription factor to control it?
That would be a good one. We haven't. And the thickness of the lines are correlated with how many peptides was identified. Right? So we use this. We use that. We also use quantitative information from our heavy and light to kind of get rid of contaminant proteins. And 18 proteins were identified in both. So if you take those proteins, for instance, if I take the proteins that were specifically pulled down in the resistant line, and I ask the Go ontology network, what are those proteins seem to be associated with, I get similar results to what I saw when I take the whole proteome. I see a set of proteins that are correlated with the unfolded protein response, glycolysis, regulation of apoptosis, and phosphorylation. Gives me confidence. If I do the opposite, if I look at proteins that were specifically enriched in the parental line, I see regulation of DNA binding. I see apoptosis, signal transduction. Okay. Very different pathways, but both that were supported by when I looked at the gross changes in the proteome. Most important, and that goes to the question, is that you always have to validate. In this case, we did independent validation, right? Biological replicates in cell lines, and we use Western blots as detection. So in this case, for instance, you look at association of June. So here's the parental line. When you give it gefitinib, when you lower the phosphorylation of EGFR, notice that C-June no longer interacts with EGFR. It's a change based on the phosphorylation of EGFR and C-June. If you look at C-June by itself, you notice that there's more C-June in the resistant one, which is what our initial discovery programs look at. And you notice some members of the unfolded protein response, PDIA4, which also is known to regulate apoptosis, interacts a lot more in the resistant line than is in the parental line. The other proteins that are interesting is MYC6, which is a known negative regulator of EGFR signaling, and AGR2. A lot more signal here than in the parental lines. Why is that important? Is because we know that MYC6 is known to bind to the cytoplasmic portion of EGFR, and it prevents its activity. There's a number of reports that have looked at levels of MYC6 and response to EGFR TKIs. This is the first case where we found an interaction with C-June and MYC6, which kind of brings MYC6 from the nucleus, cytoplasm, and to the receptor. Okay? So after we did this effort, the next question is, what's the biological relevance? So we did RNA-seq, we did gene set enrichment analysis, and we found that our signature matches a number of published reports on resistance to this drug. And we also did ChIP-seq. And in ChIP-seq, when you look at C-June ChIP-seq, we see a dramatic difference between the parental and resistant line of enrichment. So there's a big change on occupancy of the C-June promoter. So it's not just higher abundance, but it's in terms of transcriptional activity, it's much, much higher transcriptional activity. Is there a similar association between increased C-June levels and decreased EGFR in vivo? We looked at the number of publicly available databases that had phosphoproteomic data, and we noticed that changes in phospho-EGFR are linked to C-June expression. And we did ANOVA analysis. So it seems to be happening in patient samples. And if I look at patients who have activating mutations in EGFR, you would hypothesize that activity of the protein is high because you have, for instance, this mutation. If those patients have high C-June, activity seems to be low in a subset of these patients. Okay? So it seems to be correlated in a subset of patients. And if I take my signature, so here's my parental cell line, and here's a patient that is also responsive to this drug, you see that gene expression profiles seem similar, and here's a patient who is refractory to the drug, and here's our expression pattern from the resistance, and it looks qualitatively similar. So ultimately, if you put this together, notice that in the sensitive model, C-June seems to be interacting with EGFR, especially in the phosphorylation state. It promotes transcriptional activity of genes associated with EGFR. For instance, EGF is upregulated in this case. However, in the resistant one, C-June seems to be interacting with a number of known downregulators of EGFR. It promotes upregulation of glucose metabolism, unfolded protein response, and known negative regulators such as AGR2 and MYC6. Right? So very different interactome for both. So here's our conclusions. So this mechanism seems to be mediated by C-June. We're looking at more patient samples to find a bigger cohort. The macromolecular interactions are very different. The changes in the proteome seems to be very different, as you might expect by changing the transcriptional activity of a transcription factor. Okay? And it seems to be very coordinated. it seems that on one hand, it's upregulating proteins known to downregulate this pathway, and at the same time, it's downregulating genes that are known to promote activity of EGFR. So it's doing both. And so now the role of these two proteins in patients who are resistant is unknown, and that's something that someone can go and look at more specifically. So this is a big team effort. So our group leader is Dr. David Agus. We have a large number of people who work on these projects with us. We have collaborators at Fred Hutch and Stanford who contributed to this. With that, any questions?
Thank you, Dr. Kani, for your lucid presentation. It's open for discussion.
That's a very nice presentation. Now that you know the mechanism of action of broad resistance to EGFR transcriptase inhibitors, can you, as a vision as a follow-up project, find out a combination therapy that overcomes resistance? So through the knowledge you have in these, with the profiles of these proteomics, how to identify synergistic combination or some kind of drug that the patient can take to overcome this kind of resistance. So therefore, you prevent patients who are going to develop resistance from either developing resistance or once it occurs, you have an antidote, the kind of antidote
Right.
to resistance.
So we did similar work that you're describing. Actually, Shannon Momenthaler published a work where she looked at dosing with paclitaxel and EGFR tyrosine kinase inhibitors, and she modeled pulse dosing versus differential temporal kinetics, and she published that a few years ago. Right? So we did that. c-Jun inhibitors are available. In my opinion, in preclinical models, they're good. I haven't seen evidence in clinical models how well c-Jun inhibitors work. So in mouse models, I can do that experiment with you, right, to see which combination would work. Exactly. Exactly.
The other question I had is just for my personal knowledge. Do you know any commercially available cell line, cancer cell line resistance to mechanistic inhibitors such as the LGBT, or do you have to create this resistance?
I can give you mine. I can give you mine. Yeah, we have a repository of I think 20 cell lines that are either sensitive or resistant to a number of different drugs.
And do you know the kind of resistance for?
In some of them we do, right? So for instance, this one we have Shannon's published work on T790M mutations. So William Powell, I think he's at MD Anderson, has those cell lines. They're called PC9s. He would share those with you. There's also HCC 827s that have EMT as a mechanism of resistance. And there's HCC 827s that have CMET as a mechanism of resistance. So in that one cell line, there's 3, or if you count mine, 4 different mechanisms. Now, remember, my cell lines, although they're HCC 827s, are probably different because the way I've handled it, right? But yes, they're available. Yes?
Yeah, I was wondering, are you thinking of any identifying the molecule which can disrupt the CPI interaction among the factors?
Like a protein-protein inhibitor. So we're not good at that. We don't do that, but if someone would be willing to tackle that with some sort of a drug screen, that would be wonderful. I think that would be very powerful to do that. Yes.
Have you tried J&K inhibitors?
Yeah, we did. It does work. It does work, right? You would, yeah, if you knock out the kinase, that does that.
Okay. So in one slide you show that the thickness of the lines actually represent how many peptides you identify from either the APMS or the bio ID, right? Do you think it's meaningful to also because for this identification is basically identify one protein, in it. Do you think it's also meaningful to introduce quantification method for the data analysis, either
So we did that. We labeled cells with either heavy lysine
Oh,
or
yeah,
light
that
lysine.
one, yeah.
So we use that label to tell us what's a true ID versus a false ID. But you can also use the ratio between the lysines as a way to get relative intensity. So for instance, if my signal is 10-fold increased with a heavy peptide with its heat compared to that, and in terms of abundance, stoichiometry is a lot more. For me, all these methods are discovery tools, and if you find proteins that are interesting, then you really have to specifically validate with another technique.
Right.
And generally, when I did the co-ID confirm, they match the quantitation from the proteins. So I think I'm on the right track. Are there examples where it wasn't as good? Yes. But yeah, you could do those things if you're really interested in the stoichiometry of how those proteins are in those forms.
Another good thing is that for the cells, you compare the resistant one and the normal one, and for a lot of protein-protein interactions, they are like transient and very dynamic. And in that case, do you think it's more meaningful to compare like drug-treated cells versus the non-treated cells to see like what is the dynamic changes of the protein-protein interaction?
So we did that. Part of the data I presented in my talk last night was based on giving drug at 2 hours and 16 hours and looking at the change in the proteome of those particles. So we have that data. This is kind of an extension of that work where that one looked at transient changes, this looked at long-term changes.
If you look at like 12 hours, 16 hours, do you compare like the two time point or compare them to the like the control protein?
To the resistant one, yeah, we've done that. So there's some proteins that under short-term, for instance, 2 and 16 hours, initially go up, go down, and then after you get acquired resistance, depending on what protein that is, go back up or to stay down.
But if you only first select, you only have 2 labels, right? Then how can you quantify so many different samples?
Oh, so it's a separate experiment. So yeah, they're separate experiments.
I can understand.
Yeah, have you checked the endocytosis of different receptor? Maybe that's the
So in this
different
case,
cell lines.
one of the things that AGR2 does is that it changes accessibility of the receptor to the cell surface.
Oh, okay.
So it's still expressed. It's expressed at lower levels. But if you do immunofluorescence, you notice that in parental cell lines, it's all on the cell surface. But in the resistant one, it's there, but it's not on the cell surface.
Oh, okay. Then
Which means it's changing, right, the localization.
Okay. Then you have some.
So for C2 interactors, if you compare your data from the 2 types of experiments to everything that's been described in the literature, how many of them did you know of the known interactors do you re-identify and how many did you miss?
So I did that early on. I can probably share the data with you. Typically, we found, I think it was 5 to 10% of the proteins that we found were found in our sequence. One thing that was interesting for me is that interaction with C-gene with a lot of the members of the unfolded protein response was rarely reported. Now, using a lung cancer cell line that had EGFR that's limited resistance might be a complete different context comparing to anyone else who's looked at C-gene and something that can have a complete different impact. So I would take that with a grain of salt.
So 90% of the interactors are new neuroscience, not previously described. And when you say that, you're just comparing the baseline or you're comparing resistant versus non-resistant?
So my baseline in the parental was smaller. I had much smaller interactome. In the resistant one, right, so for me the baseline is the parental because that is kind of a cell line that's not artificial. The resistant one is completely new, so I don't think we very much at the interaction of a resistant line who was been published.
Right.
So I think the ideal experiment to do as a follow-up is to look at specific C-gene interactions in a series of different cell lines to see if you can find the same trends.
So when you look at the ones that are common, the KPMS and the bio-ID, you said there are 8 or 10 that are common?
Yeah, very
Is
few.
the ratio still the same?
No.
It meaning of the known versus novel? Is it still 90%, or most of them are still new?
So
And they still overlap in the two data sets?
So I didn't look at any overlap proteins that were just identified in the resistant model with proteome. But in terms of bio-ID interaction with APMS, there's two ways to look at bio-ID interactions. Most people in literature take whatever you get from the bio-ID method and you report that the same way as you do with APMS. I wanted to get an extra level of scrutiny, and in my search algorithms for the proteomics, I said I require a mass shift of $226 corresponding to the bio on any lysine. So my bio-ID interactome is much smaller than my APMS. If I don't do that, I get tons of proteins from my bio-ID. But for me, I'm really interested in proteins that are specifically biochemical.
But then you have to get those peptides that are biochemical.
That's
You're
right.
narrowing it down because you've got so many lysines and only some of them may be modified. It all depends on the proximity and the 3D confirmation.
It lowers your pool if you put a stringent search criteria that I need a peptide that has lysine biochemical. And for me, that peptide has to have a heavy lysine because it's coming from cells that was isopropyl rich with peptide.
So you decrease your pool, so that filter reduces it by what percent?
I don't know the numbers exactly, but I know generally if I don't have that filter for a bio-ID experiment, you get 300 IDs. When I add that filter, I reduce that to 15.
So very stringent.
Very stringent. So most publications of bio-IDs don't do that.
Right. I don't know anyone who does that. Is anyone doing that?
I don't know.
Yeah.
To follow up on the bio-ID, you said, I never read any literature on that, so I know pretty much nothing for your talk. So it's based on how close two proteins are with each other, right? Do you think that could be another reason why the number you identify from bio-ID is less than the APMS? Because like in the APMS, you can identify something within a complex, like two proteins are not directly interacting with each other, right?
I think it depends what your fusion is.
Okay.
Right. So, proteins that have linkers that are flexible. So the C-terminal region of the protein is floppy and is able to kind of move around. And if you put a bio-ID fusion there, my guess is you can actually bioaccumulate a lot of substrates non-specifically because it's just floating around.
Right.
So I think it's on a case-by-case basis. For my case, as we discussed, I wasn't really interested in finding a lot of proteins. I was really interested in finding the right proteins, the ones that I really knew that were interacting with this pathway. So for me, I added that extra level of stringency. So if you just want a general interaction map, I think generally what people do is just put a bioinformatic, do a very stringent pull down, run it through a graph on a number of databases, and that's your interaction map. So it's amenable to high throughput studies if you want to do it.
Thank you. Token of appreciation for your this thing. Please.
Oh, thank you.
Okay. Thank you.